ASO design and screening, a comprehensive overview. Antisense oligonucleotides, ASOs, represent a powerful class of therapeutics that have revolutionized the treatment of a variety of genetic disorders. By selectively binding to specific RNA sequences, ASOs can modulate gene expression, offering precise control over biological processes that are implicated in disease. The success of ASO-based therapies hinges on meticulous design and rigorous screening processes to ensure efficacy, specificity, and safety. The journey from conceptualizing an ASO to its therapeutic application involves several critical steps. This includes selecting the appropriate genetic target, designing ASOs with optimal chemical modifications, and employing comprehensive screening methodologies to identify the most effective candidates. Each step is underpinned by a deep understanding of molecular biology, RNA chemistry, and advanced biotechnological tools. In this comprehensive overview, we delve into the intricacies of ASO design and screening. We explore the principles of target selection, the detailed chemistry behind ASOs, and the mechanisms through which they exert their effects. Furthermore, we discuss the sophisticated screening methodologies that are essential for validating ASO function and specificity, from in silico predictions to in vitro assays and in vivo studies. By integrating these elements, researchers can develop ASO therapeutics that not only achieve high levels of efficacy, but also minimize off-target effects and adverse reactions. This article aims to provide a detailed technical roadmap for the design and screening of ASOs, equipping scientists and clinicians with the knowledge needed to harness the full potential of this transformative technology. Antisense oligonucleotides, ASOs, are short, synthetic strands of nucleic acids designed to bind to specific messenger RNA, mRNA sequences. By binding to these target mRNAs, ASOs can modulate gene expression through various mechanisms, including the degradation of the mRNA, alteration of splicing, or inhibition of translation. ASO design and screening is a critical process in the development of these molecules for therapeutic applications. This article provides an in-depth technical overview of ASO design and screening, including key principles, methodologies, and considerations. Contents: Introduction. Key principles of ASO design: target selection, gene selection, transcript isoforms, target site selection, ASO chemistry, backbone modifications, sugar modifications, base modifications, conjugation strategies, mechanism of action, RNase, H-mediated degradation, splice modulation, translation inhibition, screening methodologies, in silico screening, in vitro screening, in vivo screening, advanced screening techniques. Conclusion. Key principles of ASO design: target selection, gene selection. Identify the gene of interest based on its role in disease pathogenesis. This involves understanding the gene's function, expression pattern, and the nature of its involvement in the disease. Transcript isoforms. Consider the presence of alternative splicing isoforms of the target mRNA. Different isoforms might have different functional roles, and the ASO might need to target a specific isoform. Target selection is the first and one of the most critical steps in the design of antisense oligonucleotides, ASOs. A well-chosen target can significantly influence the efficacy and specificity of the ASO and ultimately its therapeutic potential. Here we delve into the technical aspects of target selection, covering gene selection, transcript isoform considerations, and target site accessibility. Gene selection, disease relevance, pathogenic mechanism. Understanding the pathogenic mechanism of the disease is crucial. This involves identifying genes that are directly involved in the disease process, whether through overexpression, underexpression, or mutation. Genetic studies. Utilize genome-wide association studies, GWAS, exome sequencing, and other genetic studies to identify genes associated with the disease. Functional studies. Employ techniques like RNA interference, RNAi, CRISPR, Cas9, gene editing, and knockout models to validate the role of the gene in the disease. Expression profiling, tissue-specific. Expression. Assess the expression levels of the gene across different tissues using techniques such as RNA sequencing, RNA-seq, and quantitative PCR, qPCR. This helps determine if the gene is expressed in the relevant tissues affected by the disease. Temporal expression. Investigate the expression profile of the gene over time, particularly if the disease has a developmental or progressive aspect. This can be done through time-course RNA-seq experiments. Gene function biological pathways. Map the gene to known biological pathways and networks using databases like KEGG, Reactome, and BioGrid. Understanding the gene's role in these pathways can provide insights into potential off-target effects and unintended consequences of ASO intervention. Protein function. Investigate the function of the protein encoded by the gene. This includes understanding its interactions, post-translational modifications, and involvement in cellular processes. Transcript isoform considerations, alternative splicing, splice variants. Many genes produce multiple mRNA isoforms through alternative splicing. Isoform-specific expression can be crucial for targeting the correct mRNA variant. Isoform-specific ASOs. Design ASOs that target isoform-specific exons or splice junctions to ensure the modulation of the correct transcript. Tools like the UCSC Genome Browser and Ensemble can help identify these regions. Isoform functionality. Functional isoforms. Determine the functional differences between isoforms. Some isoforms might have dominant negative effects or might be non-functional. Disease-associated isoforms. Identify isoforms that are specifically associated with the disease. This can be done through transcriptomy, analysis of disease versus healthy samples. Quantitative analysis. Isoform expression levels. Quantify the expression levels of different isoforms using RNA-seq data. This involves using software tools like cufflinks, stringtie, or Callisto to assemble and quantify transcripts. Relative abundance. Assess the relative abundance of isoforms to prioritize which isoform to target based on their contribution to the disease phenotype. Target site accessibility. Secondary and tertiary structure RNA folding. RNA molecules fold into complex secondary and tertiary structures, which can affect the accessibility of potential ASO binding sites. Predict the folding patterns using computational tools like RNA-fold or M-fold. Accessible regions. Identify accessible regions such as loops, bulges, and unstructured regions, where ASOs are more likely to bind effectively. These regions can be identified through experimental methods like SHAPE, selective 2-hydroxylacylation analyzed by primer extension, or computational predictions. RNA-protein interactions, ribonucleoprotein complexes. mRNAs often interact with RNA-binding proteins, RBPs, forming ribonucleoprotein, RNP complexes. These interactions can mask or expose potential ASO binding sites. RBP binding sites. Map RBP binding sites using cross-linking and immunoprecipitation (CLIP) techniques. Avoid targeting these regions unless the ASO is designed to disrupt the RNP interaction. Conservation analysis, cross-species. Conservation. Analyze the conservation of the target mRNA sequence across different species. Highly conserved regions are more likely to be functionally important and can provide insights into the essential nature of the target site. Phylogenetic footprinting. Use phylogenetic footprinting to identify conserved elements within the mRNA. Tools like UCSC Genome Browser's FastCons or GERP scores can aid in this analysis. Experimental validation in vitro assays. Validate target site accessibility using in vitro assays. This can include RNase H cleavage assays to test whether the ASO can recruit RNase H to the target site. Cell-based assays. Use cell-based models to assess ASO binding and activity. Techniques like RNA immunoprecipitation, RIP, or ASO pull-down assays can provide evidence of ASO binding to the target mRNA. Target selection in ASO design is a multi-step process that requires a thorough understanding of the gene's role in disease, its expression profile, the structure of its mRNA, and the accessibility of potential binding sites. By integrating bioinformatics tools, experimental validation, and functional studies, researchers can identify optimal targets for ASO therapy. This meticulous approach ensures that the ASOs designed are both effective and specific, minimizing off-target effects and maximizing therapeutic potential. Target site selection accessibility. The target site on the mRNA must be accessible for binding. This can be influenced by the secondary and tertiary structures of the mRNA, which can obscure potential binding sites. Conservation. For therapeutic applications, targeting regions of the mRNA that are conserved across species can be beneficial for cross-species efficacy studies. Target site selection is a critical step in the design of antisense oligonucleotides, ASOs. The chosen target site on the mRNA must be accessible and functionally relevant to ensure effective and specific ASO binding. Here, we delve into the technical aspects of target site selection, covering RNA secondary and tertiary structure analysis, conservation and functional relevance, accessibility prediction, and validation techniques. RNA secondary and tertiary structure. Analysis, secondary structure prediction, computational tools. Utilize computational tools such as RNA-fold, M-fold, and NUPAC to predict the secondary structure of the target mRNA. These tools use algorithms based on thermodynamic models to predict the most stable folding patterns of the RNA. Dot plots. Generate dot plots to visualize base pairing probabilities. These plots help identify regions of the mRNA that are more likely to be single-stranded and thus accessible to ASOs. Tertiary structure considerations 3D folding. Predicting the tertiary structure of RNA is more complex and often requires specialized tools like RNA Composer or iFold RNA. Understanding the 3D conformation helps identify regions that might be occluded due to higher-order structures. Experimental techniques. Use experimental techniques such as X-ray crystallography, cryo-electron microscopy, cryo-EM, or nuclear magnetic resonance, NMR spectroscopy, to obtain high-resolution structures of the target mRNA. Structure-function relationship functional domains. Identify functional domains within the mRNA, such as ribosome binding sites, splicing sites, and regulatory elements. Targeting these regions can have significant effects on mRNA function. Mutation analysis. Study disease-associated mutations to understand their impact on RNA structure and function. This can guide the selection of target sites that are crucial for the mRNA's role in the disease. Conservation and functional relevance. Conservation analysis. Cross-species. Conservation. Analyze the conservation of the target mRNA sequence across different species using tools like UCSC, genome browsers, FastCons, or GERP scores. Highly conserved regions are likely to be functionally important and are good candidates for targeting. Phylogenetic footprinting. Use phylogenetic footprinting to identify conserved elements within the mRNA. This involves aligning the mRNA sequences of different species and identifying conserved regions. Functional relevance essential regions. Target regions essential for mRNA stability, translation, or splicing. These regions are often more impactful for therapeutic intervention. Regulatory elements. Identify and target regulatory elements such as miRNA binding sites, AU-rich elements, or splicing enhancers, silencers. Disrupting these elements can modulate mRNA function. Accessibility prediction, computational predictions, RNA accessibility tools. Use tools like SFOLD, OligoWalk, or RNase-NP to predict RNA accessibility. These tools analyze the folding patterns and calculate the probability of a region being single-stranded. Thermodynamic calculations. Assess the free energy changes associated with ASO binding to different regions. Lower free energy indicates higher binding affinity and accessibility. Experimental validation. Shape mapping. Use selective 2 hydroxyl acylation analyzed by primer extension, SHAPE, to probe RNA structure and identify accessible regions. SHAPE reagents modify accessible nucleotides, and the modifications are detected by reverse transcription and sequencing. DMS footprinting. Dimethyl sulfate DMS footprinting involves treating RNA with DMS, which methylates accessible adenine and cytosine residues. The modified nucleotides are then identified using primer extension assays. Validation techniques in vitro assays. RNase H cleavage assay. Test ASO-induced RNase H cleavage in vitro. This assay confirms whether the ASO can bind to the target site and recruit RNase H to degrade the mRNA. Electrophoretic mobility shift assay, EMSA. Use EMSA to study the binding affinity of ASOs to their target mRNA. The binding is visualized by a shift in the mobility of the RNA-ASO complex during electrophoresis. Cell-based assays, luciferase reporter, assay. Construct a luciferase reporter containing the target mRNA sequence. Measure the effect of ASO binding on luciferase expression to validate target site accessibility and ASO activity in a cellular context. RNA immunoprecipitation, RIP. Perform RIP assays to pull down the target mRNA-ASO complex from cell lysates. This assay helps validate in vivo binding of the ASO to its target. In vivo validation animal models. Use relevant animal models to study the biodistribution, efficacy, and safety of ASOs targeting the selected site. This includes assessing the reduction in target mRNA and the therapeutic effect. Pharmacokinetics and pharmacodynamics, PK/PD. Conduct PK/PD studies to understand the in vivo dynamics of ASOs, including their stability, tissue distribution, and biological activity. Target site selection for ASOs involves a comprehensive analysis of RNA structure, conservation, and functional relevance. By integrating computational predictions with experimental validation, researchers can identify optimal target sites that ensure effective and specific ASO binding. This meticulous approach maximizes the therapeutic potential of ASOs by targeting the most accessible and functionally relevant regions of the mRNA. Bioinformatics tools overview technical analysis of RNA fold, M-fold, and NUPAC RNA secondary structure prediction is an essential step in understanding RNA function and designing antisense oligonucleotides, ASOs. Here, we delve into the technical details of three widely used computational tools for RNA secondary structure prediction: RNA fold, M-fold, and NUPAC. This section covers their underlying algorithms, methodologies, input-output formats, and practical applications. RNA fold overview, developed by the Vienna RNA package. Primary use: predicting RNA secondary structures based on thermodynamic stability. Algorithm and methodology dynamic programming, DP. RNA fold uses a dynamic programming algorithm to compute the minimum free energy MFE structure of an RNA sequence. The algorithm considers all possible base pairs and evaluates their contributions to the overall free energy, iterating through the sequence to build up a matrix of optimal substructures. Turner Energy Model, 1999. RNA fold relies on thermodynamic parameters from the Turner model, which provides experimentally determined free energy values for different RNA structural motifs, e.g., hairpins, internal loops, bulges. The model includes contributions from base stacking, hydrogen bonding, and loop entropy, partition function. In addition to predicting the MFE structure, RNA fold computes the partition function, which provides the probability of each base pair forming in the ensemble of possible structures. This allows for the generation of base pairing probability matrices and dot plots, offering insights into regions of structural flexibility. Input and output. Input: RNA sequence in plain text or FASTA format. Optional parameters: temperature, dangles, treatment of dangling ends, and constraints, forced pairing, unpairing. Output: dot bracket notation represents the MFE structure with paired bases, indicated by matching parentheses, and unpaired bases by dots. Free energy value: the calculated free energy of the MFE structure, base pairing probability matrix, a matrix showing the probability of each base pair. Dot plots: visual representations of base pairing probabilities and MFE structures. Practical applications: ASO design. RNA fold helps identify accessible, single-stranded regions suitable for ASO targeting, mutational analysis, predicts the impact of mutations on RNA structure and stability, RNA function studies, analyzes the structural dynamics of RNA in different conditions, e.g., temperature changes. M-fold overview, developed by Michael Zucker, primary use, predicting RNA secondary structures and suboptimal structures, algorithm and methodology dynamic programming. M-fold uses a similar dynamic programming approach to RNA-fold, constructing a matrix of optimal substructures to identify the MFE structure. The algorithm evaluates all possible base pair interactions and computes their contributions to the overall free energy. Suboptimal structures. In addition to the MFE structure, M-fold generates suboptimal structures that fall within a specified energy range of the MFE. This feature provides insights into the structural variability and potential alternative conformations, temperature dependency. M-fold allows for predictions at different temperatures, accounting for the effects of temperature on RNA folding and stability, input and output input. RNA sequence in plain text or FASTA format. Optional parameters. Folding temperature, ionic conditions, e.g., Mg2+ concentration, and constraints. Output. Dot bracket notation. MFE and suboptimal structures represented in dot bracket notation. Free energy values. Calculated free energy values for MFE and suboptimal structures. Secondary structure diagrams. Graphical representations of predicted structures. Energy dot plots. Visual representations showing the energy landscape and suboptimal structures. Practical applications. ASO design. Identifies robust target sites by evaluating structural stability across different conditions. Comparative analysis. Compares structures of wild-type and mutant RNA sequences to understand the effects of mutations. Structural studies. Explores the temperature-dependent folding dynamics of RNA. NUPAC overview. Developed by the NUPAC team at Caltech. Primary use. Predicting RNA secondary structures and analyzing RNA-RNA interactions. Algorithm and methodology, multi-state ensemble model. NUPAC treats RNA molecules as ensembles of multiple states, each contributing to the overall partition function. The algorithm computes the partition function for RNA sequences, allowing for the prediction of a range of possible structures and their probabilities. Complex formation. NUPAC excels in predicting the secondary structures of RNA complexes involving multiple interacting strands. It models RNA-RNA interactions and computes the equilibrium base pairing probabilities for the complex. Thermodynamic model. The tool uses a comprehensive thermodynamic model that includes parameters for base stacking, loop formation, and other structural features, similar to the Turner model, but extended for multi-strand interactions. Input and output. Input. RNA sequence SS in plain text or FASTA format. Optional parameters. Complex formation scenarios, temperature and ion concentrations. Output dot bracket notation. Predicted secondary structures for individual RNA molecules and complexes. Base pairing probability matrices. Matrices showing the probability of each base pair in the ensemble of structures. Free energy values. Calculated free energy values for the predicted structures and complexes. Ensemble defect metrics. Measures of structural fidelity and defects in the predicted ensemble. Practical applications. ASO design. NUPAC's ability to predict RNA complexes is useful for designing ASOs that target structured regions or disrupt RNA-RNA interactions. Synthetic biology. Used to design RNA molecules with specific structural and functional properties such as RNA switches and ribozymes. Interaction studies. Analyzes RNA-RNA interactions in complex biological systems. Comparative analysis. Algorithmic strengths. RNA fold. Excellent for probabilistic analysis and identifying single-stranded regions with high base pairing probabilities. M-fold. Provides insights into structural variability with suboptimal structures and temperature-dependent predictions. NUPAC. Superior for modeling RNA complexes and multi-strand interactions, practical considerations, accessibility identification. RNA fold and NUPAC provide base pairing probabilities, aiding in the identification of accessible target sites for ASOs. Stability and variability. M-folds, suboptimal structures, and temperature-based predictions offer valuable insights into structural robustness, complex interactions. NUPAC's ability to predict RNA complexes is crucial for designing ASOs that disrupt RNA-RNA interactions. RNA fold, M-fold, and NUPAC are powerful tools for RNA secondary structure prediction, each with unique strengths and applications. RNA fold excels in probabilistic predictions and identifying accessible regions. M-fold offers insights into structural variability and stability, and NUPAC is ideal for studying complex RNA interactions. In ASO design, these tools collectively provide a comprehensive understanding of RNA structure, facilitating the selection of optimal target sites for therapeutic intervention. ASO chemistry backbone modifications. Common modifications include phosphorothioate (PS) linkages, which improve nucleus resistance and binding affinity. Sugar modifications, 2-O-methyl, 2-O-ME, and 2-O-methoxyethyl, 2-MOE modifications enhance binding affinity and reduce toxicity. Base modifications, locked nucleic acids, LNAs, and other modifications can be used to increase binding affinity and specificity. The chemistry of ASOs is crucial for their stability, binding affinity, specificity, and overall therapeutic efficacy. This section delves into the technical details of ASO chemistry, covering backbone modifications, sugar modifications, base modifications, and conjugation strategies. Backbone modifications. Backbone modifications are critical for enhancing the stability and pharmacokinetic properties of ASOs. The most common modifications involve replacing the natural phosphodiester linkages with more stable alternatives. Phosphorothioate (PS) modification structure. In PS linkages, a non-bridging oxygen atom in the phosphodiester bond is replaced with a sulfur atom. Benefits: nucleus resistance. The sulfur atom provides increased resistance to nucleus degradation, enhancing ASO stability in biological fluids. Enhanced binding. PS linkages improve the binding affinity of ASOs to plasma proteins, which can increase their half-life in vivo. Cellular uptake. PS-modified ASOs exhibit better cellular uptake compared to unmodified ASOs. Considerations. PS modifications can sometimes reduce binding specificity and may introduce off-target effects due to altered interactions with proteins. Phosphorodimidate morpholino oligomers, PMOs. Structure. PMOs have a backbone where the ribose sugar is replaced with a morpholine ring and the phosphodiester linkage is replaced with a phosphorodimidate group. Benefits. High stability. PMOs are highly resistant to nucleases and other enzymatic degradation. Reduced toxicity. PMOs generally exhibit low toxicity in vivo. Considerations. PMOs have different charge properties compared to natural oligonucleotides, which can affect their pharmacokinetics and cellular uptake. N3P5 phosphoramidate structure. This modification involves a linkage between the nitrogen atom at the 3 position and the phosphorus atom at the 5 position. Benefits: nucleus resistance, enhanced stability against nucleus degradation, improved affinity. These modifications can improve binding affinity to the target RNA. Considerations: the synthetic complexity and potential toxicity need careful evaluation. Sugar modifications. Modifications to the ribose sugar in ASOs can enhance their binding affinity, stability, and reduce off-target effects. 2-isomethyl, 2-iso-ME structure. A methyl group is added to the 2-isen hydroxyl group of the ribose sugar. Benefits: enhanced stability, increased resistance to nucleus degradation, increased binding affinity, improved hybridization affinity to the target RNA, reduced immunogenicity, lower potential for immune activation compared to unmodified oligonucleotides. Considerations. These modifications can affect the overall conformation of the RNA ASO complex. 2-O-methoxyethyl, 2-MOE, structure. A methoxyethyl group is attached to the 2E hydroxyl group. Benefits: high affinity, further enhances binding affinity to the target RNA compared to 2-OMe modifications. Nuclease resistance, provides significant resistance to nucleases, improved pharmacokinetics. Better pharmacokinetic profile due to increased stability and affinity. Considerations: increased hydrophobicity can affect solubility and distribution in biological systems. Locked nucleic acids, LNAs. Structure. LNAs contain a methylene bridge that locks the ribose sugar in a rigid C3 endo conformation. Benefits: high affinity. LNAs exhibit significantly higher binding affinity to complementary RNA sequences. Stability. Enhanced resistance to nucleases and other degradative enzymes. Considerations. LNAs can increase the overall rigidity of the ASO, which might impact its ability to interact with target RNA in dynamic cellular environments. Base modifications. Base modifications can enhance the specificity and binding affinity of ASOs by altering the chemical properties of the nucleobases. Peptide nucleic acids, PNAs. Structure. PNAs have a peptide-like backbone where the nucleobases are attached to a polyamide chain. Benefits: high affinity. PNAs bind very tightly to complementary DNA and RNA sequences, nuclease and protease resistance, highly resistant to enzymatic degradation. Considerations. PNAs are neutral molecules, which can affect their solubility and cellular uptake. Modified nucleobases examples: pseudoridine, 5-methylcytosine, and 2-thioridine. Benefits: enhanced stability. Some modified bases can improve the stability of the ASO-RNA duplex. Reduced immunogenicity. Modified bases can reduce the likelihood of immune system recognition. Considerations. The introduction of modified bases needs to be carefully balanced to avoid disrupting the overall structure and function of the ASO. Conjugation strategies. Conjugating ASOs to various molecules can enhance their delivery, targeting, and efficacy. Lipid conjugates examples: cholesterol, tocopherol. Benefits: enhanced cellular uptake. Lipid conjugation improves ASO uptake by cells, particularly through endocytosis. Improved biodistribution. Lipid conjugated ASOs can have better distribution in target tissues. Considerations. Lipid conjugates can affect the solubility and potential off-target interactions of ASOs. Peptide conjugates examples: cell-penetrating peptides, CPPs, receptor-targeting peptides. Benefits: targeted delivery. Peptides can facilitate targeted delivery to specific cell types or tissues. Enhanced uptake. CPPs can significantly increase the cellular uptake of ASOs. Considerations. Peptide conjugates can introduce immunogenicity and potential toxicity. Antibody conjugate structure. ASOs conjugated to antibodies or antibody fragments. Benefits. Specific targeting. Allows for highly specific targeting to cells expressing the corresponding antigen. Reduced off-target effects. Increases the specificity and reduces systemic exposure. Considerations. The large size of antibody conjugates can affect pharmacokinetics and biodistribution. The chemistry of ASOs is a sophisticated field that involves various modifications to the backbone, sugar, and bases, as well as the use of conjugation strategies to improve their therapeutic properties. Backbone modifications like phosphorothioate and PMOs enhance stability and cellular uptake. Sugar modifications, including 2-OMe, 2-MOE, and LNAs, improve binding affinity and resistance to nucleases. Base modifications such as PNAs and modified nucleobases can increase specificity and reduce immunogenicity. Conjugation strategies involving lipids, peptides, and antibodies facilitate targeted delivery and cellular uptake. Understanding and optimizing these chemical modifications is crucial for developing effective and safe ASO-based therapies. Mechanism of action: RNAs, H-mediated degradation. ASOs can recruit RNase H to degrade the target mRNA upon binding. Splice modulation. ASOs can bind to splicing motifs to alter pre-mRNA splicing and generate alternative splicing isoforms. Translation inhibition. ASOs can block ribosome assembly or progression, thereby inhibiting translation of the target mRNA. The mechanism of action, MOA, of antisense oligonucleotides, ASOs, involves multiple biochemical pathways through which these molecules influence gene expression. These pathways include RNase H-mediated degradation, splice modulation, and translation inhibition. Additionally, the secondary and tertiary structures of the target RNA play crucial roles in determining the effectiveness of ASO binding and activity. This section delves into these mechanisms in detail, incorporating the significance of RNA structure. RNase H-mediated degradation mechanism overview. Target binding. ASOs hybridize with their complementary target mRNA sequences through Watson-Crick base pairing, recruitment of RNase H. The ASO mRNA duplex is recognized by RNase H, an enzyme that specifically degrades the RNA strand of RNA-DNA hybrids. Detailed steps, binding and duplex formation. ASOs bind to the target mRNA, forming a stable RNA-DNA hybrid. RNase H activation. RNase H binds to the RNA-DNA hybrid and cleaves the RNA strand at multiple sites within the hybrid. Cleavage and degradation. The cleaved RNA fragments are further degraded by cellular exonucleases, leading to a decrease in target mRNA levels and subsequently the corresponding protein levels. Influence of RNA secondary and tertiary structures. Secondary structure. The secondary structure of the target RNA, including hairpins, loops, and bulges, can affect ASO binding. ASOs must be designed to target accessible single-stranded regions. Tertiary structure. The three-dimensional folding of RNA can further occlude potential binding sites. High-resolution techniques like shape and computational tools can predict these structures, aiding in the design of effective ASOs. Considerations and optimization. ASO design. Optimal ASOs are typically 15-20 nucleotides long and target single-stranded, accessible regions of the mRNA. Chemical modifications such as phosphorothioate, PS, backbones enhance ASO stability and affinity while maintaining RNase H activity. Splice modulation mechanism, overview target binding. ASOs bind to specific splice sites or splicing regulatory elements on the pre-mRNA. Alteration of splicing machinery. By blocking the binding of splicing factors or regulatory proteins, ASOs modulate the splicing process, leading to the inclusion or exclusion of specific exons. Detailed steps. Exon skipping binding to splice sites. ASOs designed to bind to splice, acceptor, or donor sites block these sites, preventing their recognition by the splicing machinery. Skipping of target exon. This blockage results in the exclusion of the targeted exon from the mature mRNA, potentially restoring the reading frame or producing a truncated protein with altered function. Exon inclusion. Binding to regulatory elements. ASOs can bind to intronic or exonic splicing enhancers, silencers, modulating the binding of splicing factors, inclusion of specific exons. By altering the binding of splicing factors, ASOs can promote the inclusion of specific exons that are normally skipped. Influence of RNA secondary and tertiary structures. Secondary structure. Splice sites and regulatory elements can be hidden within stable secondary structures, affecting the binding efficiency of ASOs. Tertiary structure. The overall folding of the pre-mRNA can influence the accessibility of splicing sites and regulatory elements. ASOs must be designed to target accessible regions. Considerations and optimization. ASO design. Design ASOs to target accessible regions of the pre-mRNA near splice sites or regulatory elements. Chemical modifications. Use of 2-O-methyl, 2-OMe, or locked nucleic acids, LNAs, can enhance binding affinity and specificity, improving splicing modulation. Translation inhibition mechanism. Overview target binding. ASOs bind to the mRNA in regions critical for translation initiation or elongation, blocking ribosome assembly by binding to the 5' untranslated region, UTR, or the start codon region. ASOs can block the assembly of the ribosome on the mRNA, inhibiting elongation. ASOs binding within the coding sequence can impede ribosome progression, halting translation. Detailed steps: binding to the 5' UTR or start codon. ASOs targeting these regions prevent the binding of initiation factors and the ribosomal subunits, thus blocking translation initiation. Binding within the coding sequence. ASOs can bind to internal regions of the mRNA, physically blocking the ribosome and causing premature termination of translation. Influence of RNA secondary and tertiary structures. Secondary structure. Regions involved in translation initiation are often structured. ASOs must target accessible single-stranded regions within these structured domains. Tertiary structure. The higher-order structure of mRNA can impact ribosome binding and progression. Effective ASOs must be designed to bind regions that are not occluded by tertiary interactions. Considerations and optimization. ASO design. Target regions critical for translation initiation or elongation that are accessible in the mRNA structure. Chemical modifications. Modifications like 2-AO-methoxyethyl, TOTA2-MOE, or peptide nucleic acids, PNAs, can increase the stability and binding affinity of ASOs, enhancing translation inhibition. Influence of secondary and tertiary RNA structures. RNA molecules fold into complex secondary and tertiary structures that can significantly impact the effectiveness of ASOs. Understanding these structures is crucial for designing effective ASOs. Secondary structure, hairpins and stem loops. These structures can hide potential ASO binding sites. ASOs must be designed to target loops or bulges where the RNA is single-stranded. Bulges and internal loops. These are typically more accessible and can serve as effective ASO target sites. Computational prediction. Tools like RNA-fold and M-fold can predict secondary structures, helping identify accessible regions. Tertiary structure. Three-dimensional folding. Higher-order interactions can occlude binding sites, making some regions inaccessible to ASOs. Techniques for analysis. Methods like shape, selective 2-or hydroxyl acylation analyzed by primer extension, and cross-linking techniques can provide information about the tertiary structure and help in designing ASOs that target accessible regions. The mechanism of action of ASOs involves complex interactions with target RNA molecules, influenced significantly by the secondary and tertiary structures of the RNA. Understanding these structures and their impact on ASO binding and activity is crucial for the effective design of ASOs. By leveraging chemical modifications and advanced computational and experimental techniques, researchers can optimize ASOs to target accessible and functionally relevant regions of the RNA, enhancing their therapeutic potential. Screening methodologies. Technical analysis of ASO. Screening methodologies. Screening methodologies for antisense oligonucleotides, ASOs, are critical in identifying effective and specific candidates for therapeutic applications. This section provides an in-depth analysis of various screening methodologies, including in silico, in vitro, and in vivo approaches, along with the tools and techniques used to evaluate ASO efficacy and specificity. In silico screening, bioinformatics tools target prediction and design BLAST. Basic local alignment search tool, BLAST, helps in identifying potential off-target effects by comparing the ASO sequence against the entire transcriptome. RNA structure, a suite of tools for predicting RNA secondary structures and assessing the accessibility of potential ASO binding sites. OligoWalk predicts the binding affinity of ASOs to target RNA sequences and evaluates off-target binding. Thermodynamic predictions, free energy calculations. Tools like RNA-fold and NUPEC can calculate the free energy of ASO-RNA hybridization, helping identify the most stable and accessible binding sites. Binding affinity. Assessing the binding affinity of ASOs to their target sequences using computational models helps prioritize candidates with the highest potential efficacy. Off-target analysis sequence similarity searches use tools like BLAST or Bowtie to identify sequences in the transcriptome that have high similarity to the ASO, which could lead to off-target effects. Cross-reactivity prediction. Evaluate the potential for ASOs to bind to unintended RNA sequences based on sequence complementarity and predicted secondary structures. In vitro screening cell-free systems RNase H cleavage assays procedure. Incubate target RNA with the ASO in the presence of RNase H and analyze the cleavage pattern using gel electrophoresis or capillary electrophoresis. Outcome. Successful ASOs will induce cleavage of the target RNA, confirming their ability to recruit RNase H. Electrophoretic mobility shift assay EMSA procedure. Mix the ASO with the target RNA and analyze the complex formation by gel electrophoresis. Outcome. The shift in RNA migration indicates binding of the ASO to the target RNA. Cell-based models reporter gene assays procedure. Construct a reporter plasmid with the target RNA sequence upstream of a reporter gene, e.g. luciferase. Transfect cells with this plasmid and treat with ASOs. Outcome. Measure reporter gene expression to assess the impact of ASO binding on target RNA function. Quantitative PCR, qPCR procedure. Treat cells with ASOs and extract RNA. Use qPCR to quantify the levels of target mRNA. Outcome. A decrease in target mRNA levels indicates effective ASO-mediated degradation or splicing modulation. Western blotting procedure. Treat cells with ASOs, lyse the cells, and analyze protein levels by Western blotting. Outcome. Reduction in target protein levels confirms the efficacy of the ASO at the translational level. RNA immunoprecipitation, RIP procedure. Immunoprecipitate RNA binding proteins, RBPs, that interact with the target mRNA followed by treatment with ASOs. Outcome. Assess changes in the interaction between RBPs and the target RNA upon ASO treatment, indicating functional disruption. In vivo screening animal models, pharmacokinetics, PK, and pharmacodynamics, PD procedure. Administer ASOs to animal models and collect samples at various time points to analyze ASO distribution, stability, and target engagement. Outcome. PK studies provide information on ASO absorption, distribution, metabolism, and excretion, ADME, while PD studies assess the biological effects of ASOs on target gene expression. Biodistribution studies procedure. Label ASOs with fluorescent or radioactive tags and administer them to animals. Use imaging techniques, e.g. fluorescence imaging, PET, to track ASO distribution. Outcome. Determine the tissue distribution and accumulation of ASOs to ensure they reach the target tissues at therapeutic concentrations. Efficacy studies procedure. Administer ASOs to disease models, e.g. mouse models of genetic disorders, and assess phenotypic outcomes. Outcome. Evaluate the therapeutic effect of ASOs on disease-related symptoms and biomarkers. Toxicity and safety assessment, acute and chronic toxicity, studies procedure. Administer single or multiple doses of ASOs to animals and monitor for signs of toxicity, e.g. weight loss, behavioral changes, organ damage. Outcome. Identify potential toxic effects and establish the maximum tolerated dose, MTD. Immunogenicity studies procedure. Administer ASOs to animals and measure immune response markers, e.g. cytokine levels, antibody production. Outcome. Assess the potential for ASOs to induce an immune response which could limit their therapeutic use. Advanced screening techniques, high-throughput screening, HTS, automated systems. Use robotic systems to conduct large-scale screening of ASO libraries against multiple targets. Multiplex assays. Implement multiplex assays to simultaneously measure the effects of multiple ASOs on different targets in a single experiment. Next-generation sequencing, NGS, RNA-seq. Use RNA sequencing to comprehensively analyze the transcriptome of cells treated with ASOs, identifying both intended and off-target effects. ClipSeq. Cross-linking and immunoprecipitation followed by sequencing to map the binding sites of RNA-binding proteins and assess the impact of ASOs on these interactions. CRISPR-based screening, CRISPR interference, CRISPR. Use CRISPR to selectively inhibit the expression of genes involved in the ASO response, identifying pathways and factors that influence ASO efficacy and specificity. CRISPR activation, CRISPR. Use CRISPR to enhance the expression of target genes, providing insights into the regulatory networks affected by ASOs. The screening of ASOs involves a combination of in silico, in vitro, and in vivo methodologies to identify effective and specific candidates. In silico tools aid in the initial design and off-target prediction. In vitro assays validate ASO binding, activity, and specificity using cell-free systems and cellular models. In vivo studies in animal models provide crucial information on the pharmacokinetics, biodistribution, efficacy, and safety of ASOs. Advanced techniques like high-throughput screening, next-generation sequencing, and CRISPR-based methods further enhance the screening process, enabling the development of highly effective and safe ASO therapies. Conclusion: Antisense oligonucleotides, ASOs, have emerged as a versatile and potent tool in the therapeutic arsenal against a range of genetic disorders. The meticulous process of ASO design and screening is paramount to their success, encompassing the careful selection of genetic targets, sophisticated chemical modifications, and comprehensive evaluation of efficacy and safety. Throughout this article, we have explored the critical components of ASO development. The journey begins with the strategic selection of targets based on a deep understanding of disease mechanisms and RNA biology. The next step involves the intricate design of ASOs, incorporating backbone, sugar, and base modifications to enhance stability, binding affinity, and specificity. We also examined the multifaceted mechanisms of action through which ASOs modulate gene expression, including RNase H-mediated degradation, splice modulation, and translation inhibition. Screening methodologies play a crucial role in identifying the most promising ASO candidates. In silico tools facilitate the initial design and prediction of off-target effects, while in vitro assays validate ASO binding and activity in controlled environments. In vivo studies in animal models provide essential insights into pharmacokinetics, biodistribution, efficacy, and safety. Advanced techniques such as high-throughput screening and next-generation sequencing further refine the selection process, ensuring that only the most effective and specific ASOs advance to clinical development. The comprehensive approach to ASO design and screening detailed in this article underscores the importance of integrating molecular biology, chemistry, and biotechnology. By adhering to these rigorous standards, researchers and clinicians can develop ASO therapies that not only achieve high levels of efficacy, but also minimize off-target effects and adverse reactions. As the field continues to evolve, ongoing advancements in RNA biology and biotechnological tools will undoubtedly enhance the precision and effectiveness of ASO-based therapies. This comprehensive overview serves as a technical guide, equipping scientists and clinicians with the knowledge and methodologies necessary to unlock the full potential of antisense oligonucleotides in combating genetic diseases.